Subtopic 3.4 Process of absorption and transportation of digested food and defecation. So observe the surface of the bare towel in photo 3.11. What can you see? Observe the three projections on the surface of the towel. Okay, so look at the surface of this towel. So this actually to allow the absorption of water. Okay, so like the towel, the wall of our small intestine has millions of fine projections called villi. Okay, so this increases the surface area for the process of absorption of digested food. Okay, digested food, which is made up of tiny molecules, is easily absorbed into the blood circulatory system through the walls of the small intestine. Okay, so these tiny molecules are then taken to every part of the body. Okay, so next figure shows the structure of a villus and the absorption of digested food in the small intestine. The inner surface of the small intestine has many villi. So the villi increase the surface area for the absorption of digested food. Absorption is the movement of digested food into the bloodstream. Okay, so this is wall of small intestine. Okay, and this thin wall of villus. Okay, so this villus. And in the villus, there are blood capillaries and lacteal. The surface area of the small intestine has many folds to add to the surface area and increase the absorption rate of the products of digestion. The wall of the villus is very thin. Okay, it is 1 cell thick to increase the rate of absorption. So the function of the blood vessels in the small intestine is to transport nutrients to all parts of the body. So this villi on the walls of the small intestine. So this is one villus. Okay, small size nutrients that can go through the wall of the small intestine are transported by the blood to the cells. Okay, so here blood capillaries. Okay, so undigested food that is larger cannot go through the small intestine wall. Okay, so it will be removed as feces. Okay, so this wall of the villus. Okay, lacteal. Okay, lacteal absorbs digested fats in the villi of the small intestine. Okay, the lacteal transfers it into the lymphatic system and then to the blood circulatory system. Now refer experiment 3.1 from your textbook. Okay, so the aim for this experiment is to study the absorption of glucose through a whisking tube. Okay, so the problem statement for this experiment: can glucose diffuse through a whisking tube? Okay, do you remember? Okay, glucose is the end product of carbohydrate. Okay, so it's the simpler molecules, okay, for the carbohydrate. So it's supposed to be can readily absorbed by our small intestine wall. Alright, so hypothesis: okay, glucose can diffuse through a whisking tube. Okay, whisking tube is actually have semi-permeable membrane. Okay, so it can let the small molecule pass through it. Okay, but bigger molecule cannot pass through it. Okay, for example, the starch, okay, which is not digested, it cannot pass through whisking tube. Okay, but glucose actually can pass through it. Okay, so the whisking tube in this experiment represents the small intestine. Okay, and then the distilled water here represents our blood. Okay, our blood in the body. Okay, so look at the variables here. Okay, constant variables, okay, or fixed variables. Okay, the type and size of whisking tube. Okay, and then temperature and time. Okay, manipulated variable. Okay, the content in the whisking tube. Okay, because we have two whisking tube. Okay, we have P and Q. Okay, this is a boiling tube and there are whisking tube with the different content. Okay, and then responding variable, the presence of glucose in the distilled water. Okay, so as you can see, materials use starch suspension. Okay, 1% of starch. Okay, and then glucose solution, whisking tube, iodine solution, Benedict solution, and distilled water. Okay, iodine solution is actually to test the presence of starch, okay, which is carbohydrate. Okay, Benedict solution is actually to detect the presence of glucose, right? Okay, so iodine solution is brown color. Okay, Benedict solution is blue color, right? Okay, so apparatus, boiling tubes, beaker, test tube, Bunsen burner, okay, stopwatch, tripod stand, wire gauge, and thread. Okay, so procedure. Okay, first, after you set up the material and apparatus, okay, soak 2 whisking tube in water to soften them. Okay, and then tie up one end of both whisking tubes using thread. Okay, and then pour 10 ml of starch suspension into one whisking tube and 10 ml of glucose solution into the other whisking tube. Okay, tie up the other end of both whisking tubes using thread. Okay, so this method, okay, or procedure, okay, you must do it properly. Okay, make sure you tie up very tight so the solution will not go out from the whisking tube from the both end of the whisking tube. Okay, okay, we only want the molecule of starch or glucose pass through the membrane of the whisking tube, not from the end of both whisking tubes. Okay, now rinse both whisking tubes using distilled water until clean. Okay, and then set up the apparatus as you can see in this figure. Okay, and then you carry out Benedict test and iodine test on the distilled water in boiling tubes P and Q. Okay, and then record your observations in the table. Okay, leave the apparatus for 30 minutes. Okay, then repeat step 7. Okay, for this experiment, okay, you need to conduct food test. Okay, as you can refer the previous activity, the food test experiment or the food test activity. So there are 2 food tests that you need to conduct in this experiment. Okay, iodine test and Benedict test. Alright, okay, but you need to conduct this test 2 times. Okay, beginning of the experiment and at the end of experiment. After 30 minutes, you need to repeat the step 7. Okay, and then record the observations. Okay, now I want to show you, okay, the iodine test. Okay, iodine test, okay, if the test is positive, okay, means if iodine can detect the presence of starch, okay, the brown color iodine will turn dark blue or blue black. Okay, so this shows the presence of starch. Okay, now for Benedict's test, okay, so this is negative reaction, means the Benedict remain blue color. Okay, if there are sugar or glucose in the solution, okay, it will turn brick red precipitate. Okay, so this brick red precipitate is formed. Alright. Okay, if negative test, so this is the color of Benedict. Alright, so this is the answers. Okay, because if you conduct this experiment, okay, it needs more than 1 hours. Okay, it more than 1 hour. Okay, because you need to conduct food tests. Okay, 2 times. Okay, and then you need to wait for this experiment another 30 minutes, so not enough time. So for boiling tube P, okay, beginning of experiment, okay, remains brown. Okay, Benedict test remains blue. Okay, so beginning experiment means no starch, no glucose, okay, presence in distilled water. Okay, end of experiment also the same for boiling tube P. Okay, and then for boiling tube Q, okay, glucose solution, okay, for iodine test means test for starch means no changes. Okay, beginning and end of experiment. Okay, but Benedict test for boiling tube Q, beginning of experiment, no glucose in the distilled water. Okay, but after experiment, after 30 minutes, okay, after we conduct the food test, okay, so we can see the Benedict test change, okay, to brick red precipitate or brick red precipitate is formed. Okay, it shows there are glucose in distilled water in boiling tube Q after 30 minutes. Okay, so the discussion. Okay, number 1. Okay, what is represented by the whisking tube and distilled water in the beaker? Okay, so in this experiment, whisking tube represents our small intestine. Okay, and then distilled water represents our blood. Okay, 2 precautionary steps, okay, that need to be taken when carrying out this experiment. Okay, so number 1, we need to tie the whisking tube properly. Okay, and then second, okay, you need to rinse the whisking tube after you tied, before you insert into the distilled water in the boiling tube. Okay, now inference. Okay, inference is actually the observation and you give the reasons. Okay, so in boiling tube P, okay, did you see any changes in boiling tube P? Alright, so the iodine solution remains brown, okay, because the molecule of starch is bigger and cannot pass through the whisking tube in the distilled water. Right, so boiling tube Q, okay, the inference for Q, okay, the Benedict solution form brick red precipitate, okay, with the presence of glucose, which is smaller size that can pass through the whisking tube in the distilled water. Okay, now the conclusion, okay, for this experiment. Okay, so the conclusion, the starch molecule cannot pass through the whisking tube as it's bigger than glucose molecule. Okay, while glucose molecule, which is smaller molecule, can diffuse through the whisking tube. Okay, so you use your own word. Okay, now we go to the process of transporting the products of digestion. Okay, food that is digested and absorbed into a villus needs to reach the cells of the body. So the molecules that are absorbed into the villus will undergo assimilation. Okay, assimilation is the process of distributing the end products of digestion for the use of the cells in our body. So our body uses the end products of digestion as follows. Okay, so glucose is used to produce energy. Okay, glucose is the end product of carbohydrate. So to produce energy, to provide energy. Okay, amino acid is used to form component of cells. Okay, means to build up our cells. Okay, amino acid from protein. Okay, fatty acid and glycerol, okay, combine to form fat, okay, which is used as heat insulator and to protect internal organs. Okay, so it warms our body and we keep our body temperature 37 degrees Celsius. Okay, all these three systems, okay, work together to ensure the digested food molecules reach the cells of the body. So digestive system, okay, breaks down large and complex particles of food into small and simple molecules so that they can be absorbed into the villi. Okay, and blood circulatory system, okay, transports small and simple molecules to the cells of the body. Okay, and then assimilation, okay, end products are distributed for formation of new cells, for respiration and regulation of body temperature.